Good morning, everyone. This is Dr. Tefide Kizildeniz from Niğde Ömer Halis Demir University, from Faculty of Agricultural Science and Technologies, the Department of Biosystem Engineering. Today, I am going to present you the climate change related factors simulations on plants. Since I have studied the master degree and my PhD degrees, I'm working in effects of climate change on the crops. So therefore, I am going to express today the different methods and how we are simulating the climate change related factors on the crops. Climate change is an effect of human activities that is a major ecological, social, economic problem of the planet. Its effects increasing temperatures, widespread melting of snows and ice, rising global sea levels, decreasing rainfall, changing species distribution, plant vulnerability and mortality, affecting crops and etc. And climate change is associated directly and indirectly to elevated CO2 temperature increase and ultraviolet radiation. This figure is representing the emissions of main greenhouse gases and their concentration in the atmosphere. As we can observe here, the carbon dioxide is the main greenhouse gases, and then methane, and then nitrous oxide and fluorinated gases. In this figure, we can observe the evolution of the atmospheric carbon dioxide concentration in the last 50 years. As we can see here, it is increasing constantly, and the fractions are the seasonals. Here we can observe the evolution of the atmospheric carbon dioxide concentration and the temperature during the last 400,000 years, according to ice cores from the Antarctica. As we can observe here, the carbon dioxide was around 280 ppm from the pre-industrial periods, and nowadays it's already reached 400 ppm. With the increasing of atmospheric carbon dioxide, we can also observe the increase of temperature. Direct effects of elevated CO2 in short time period is increasing photosynthesis and growth. In the long time period, it's affecting as a decline of photosynthesis, and this causes acclimation phenomenon. Indirect effect of elevated CO2 is the increasing of temperature, plant metabolism, and plant growth. In the cold areas, the temperature can be close to optimum temperature of growth. In the warm areas, temperature increase can exceed the optimum temperature and affect physiological processes. And it is changing the duration of seasons and phenology. Accounting for less than 0.5% UVB radiation is a minor fraction of the solar radiation reaching the Earth's surface. This is due to the action of the stratospheric ozone layer. The extinction coefficient of ozone increases several orders of magnitude as wavelength decreases. This property confers to the stratospheric ozone layer the role of protecting Earth's life form solar most harming UV radiation wavelengths. Stratospheric ozone sustains a cycle in which three forms of oxygen are involved. Molecular oxygen photodissociates into two oxygen radicals in a response to solar radiation around 240 nanometer. Oxygen radicals react spontaneously with oxygen molecules to produce ozone, which absorbs UV radiation between 200 and 350 nanometer, which reaction leads to the splitting of ozone into a molecule of oxygen and an atom of oxygen. This atom of oxygen may form ozone newly or react with another oxygen radical to produce molecular oxygen and close to the cycle. Substances such as chlorofluorocarbon compounds are dissociated by UV radiation, releasing chlorine atoms. These act as catalysts, consuming several ozone molecules before they exit the stratosphere. Due to the anthropogenic release of ozone-depleting substances, stratospheric ozone layer has been thinned at a rate of 4% per decade up to the late 1980s. The successful implementation of the Montreal Protocol contributed to reduce to a great extent the emission of ozone-depleting substances. However, due to the resilience of ozone-depleting substances, ozone levels may not recover until 2050. Ozone depletion simulations reveal the relevance of the success of Montreal Protocol. Under any scenarios of constant chlorofluorocarbon compounds emissions, UV levels would have increased several times before the end of the present century, which would have been devastating for plant life. Plant responses to UVB can be classified into stress responses and photomorphogenic responses, usually coinciding with exposures to high and low fluent rates of UVB radiation, respectively. The plant's genotyping characteristics, for example, constitutive levels of UV shielding compounds, and their previous acclimation are important factors determining whether a plant is UVB stressed or not. Usually, plants growing within their natural distribution area acclimated to UVB radiation. The objective of this work demonstrating to climate change simulation study advance on plants for individual and multiple climate change related abiotic factors. Open-top chambers, or we can call them OTC, are widely used to study the effects of elevated CO2 and other atmospheric gases on vegetation. They are plastic enclosures with an open top constructed of an aluminum frame covered by panels of polyvinyl chloride plastic film. Air is pulled into the bottom of the chamber, enriched with CO2, and then blow through the open top of the chamber. They are relatively inexpensive to construct and maintain. However, they are not appropriate for the study for large vegetations. Another study can be called spruce and peatland response under climatic and environmental change that can cause spruce. This is the modified OTC's manipulation for evaluating the response of the existing biological communities to a range of warming levels from ambient to +9 centigrade degree, provided while large under the ambient and +9 centigrade degree warming with high CO2 conditions that can be ranged between 800 to 900 ppm. The experiment provides a platform for testing mechanisms controlling the vulnerability of organisms, biochemical processes, and ecosystems to climate change. CLIMEX, or the name, another name, the climate change experiments, uses a site with an entire undisturbed boreal forested catchments enclosed within an existing very large scale greenhouses. For instance, CLIMEX was applied into the projects targeted to experimentally enrich with CO2 and raise the temperature at 2 entire forested. headwater ecosystems in order to quantify the ecosystem impacts of future climate changes and to provide essential data for the construction, calibration, and validation of process-oriented models, which is to be used to predict the response of forest and freshwater in Europe to future changes in atmospheric CO2 and climate. CLIMEX is focused on the ecosystem response to climate change, in particular the plant soil-water linkages and processes. Plant physiology, nutrient cycling, turnover of organic matter, soil and soil solution, hydrological flow paths, and runoff water quality will be investigated in these projects. Another climate change simulation experiments is the free air carbon dioxide enrichments that can call as FACE as well. FACE is a unique platform for investigating how future ecosystems are likely to respond to a higher carbon dioxide concentration without disturbing various ecosystem scale interactions. As all systems has their own advantages and disadvantages, FACE systems are offered to the researchers to study in natural aerial environment, uncontrolled humidity, wind, sunlight, and temperature, and unlimited natural rooting development, allowing to study to the taller plants. Easily available field data for both control and treated plants with low budget requirements, while other outdoor systems are not able to provide all these benefits to the researchers. Nevertheless, when other climate change related factors become an issue, these benefits turn into disadvantages. Another climate change simulation experiments is temperature gradient greenhouses. The biggest advantage of temperature gradient greenhouses is permitting to study interactions of several climate change related factors in the one experiment at the same time. For instance, coaction of temperature, air carbon dioxide level, and nitrogen fixed bacterial rays on alfalfa crops, and coaction of temperature, air carbon dioxide level, and water scarcity on grapevines were studied in these temperature gradient greenhouses. How these TGGs are working, they have there are four different greenhouses, and each greenhouse there are 3 modules. The first module has ambient temperature, and the other module, the last module, is high temperature. And there's air flux inside the greenhouses, and this air flux is providing the hot air wave. So therefore, the last module can be increase the high temperature. And two of these greenhouses having high C2 carbon dioxide concentration, while the other 2 are having ambient carbon dioxide concentrations. And also inside the greenhouses, water availability can be controlled, as you can see inside the pictures, with water sensors. Another climate change simulation methods is the growth chamber greenhouses experiments that can call GCG. Apart from the above mentioned systems for simulating climate change scenarios, one of the most important factors simulation is still missing to apply. Any of them do not allow to simulate UV radiation. GCG makes possible to apply more environmental variables within their limited workspace. One of them is carbon dioxide enrichment, another one is high temperature, the third one is water scarcity, and the last one is UVB light with additional resource of UVB radiation light. These light resources are allow GCGs to use as greenhouse or growth chamber with their 2 alternative light sources of photosynthetic active radiation. When GCGs are used as greenhouse, Due to their aluminum and glass constructions, solar UVB light is filtered and do not allow to affect subjected plants. However, foreseen climate change scenarios over the next decade counted in increased UVB radiation levels in consequence of cloud patterns fluctuations. And therefore UVB light is also possible to simulate inside the GCGs in different intensity when it is required to stimulate. Another climate change simulation experiment methods is the free air temperature increase. We can call them FATI. For simulating a climate change scenario in realist field conditions, a technique counterpart to phase was generated by Nice et al. 1997 and that named as FATI. That is attributed to adopt infrared radiation and enhances the temperature that is caused to more water loss at more leaf temperature levels in a controlled limited vegetation growth in the field conditions without enclosing the crops. Recently, FATI studies are conducted with a combination of biotic and abiotic stress factors. In conclusion, the advantages of growth chambers with rare operating dimensions offered by GCGs, allowing them to be used as greenhouses without the separating panels while keeping the same high level of environmental variables control like growth chambers. The function and application of TGGs are to examine impact of raised carbon dioxide, raised temperature and water accessibility detachedly or in collaboration. From this specialized perspective, TGGs are near work as development chambers, yet with the research-oriented greenhouses advantages. Phase systems are offered to the researchers to study in a natural aerial environment and unlimited natural rooting development, allowing to study to the taller plants easily available field data for both control and treated plants with low budget requirements, while other outdoor systems are not able to provide all these benefits to the researchers. Nevertheless, when other climate change related factors become an issue, these benefits turn into disadvantages. Plants are clearly moved away from their normal growing circumstances in growing chambers and greenhouses. Quantifying these in climate change scenarios is problematic since plants in the field condition several biotic and abiotic stress factors, and currently how these stress factors will interact with air, carbon dioxide, water availability, UVB radiation, and high temperature are still unknown. Plant research methodologies combined with climate change related factor simulations, on the other hand, are useful techniques since the former allows comparing present circumstances to close future. TGGs are a viable technique of attributing a physiological response to a single component, like as carbon dioxide, temperature, or water availability, allowing for greater mechanistic investigation into the reasons of physiological reactions to future climatic changes. However, TGGs could not cover the control of UV lights. In conclusion, in order to count up UV light effects in addition to other three climate change related factors in the site-specific climate change simulation studies on plants, the new smart system is required. I would like to thank all of you for your attention. If you have any further questions about my research as well, that I'm working in intensively temperature graded greenhouses and GCGs with collaboration of Spanish universities, you can reach to me with my email that I am leaving here. Thank you so much again, and I wish you a nice day. Bye bye.